Hello and welcome to Bio 202. This is the Heart Lecture 2, a skeletal muscle tissue review. So if you look on the left side of the screen, you can see that there is part of a skeletal muscle fiber. So a whole muscle, like your biceps muscle or your triceps muscle, is made up of these skeletal muscle fibers, or they're also called skeletal muscle cells. And you can see that on the left side it's sort of a cylindrical structure and it's covered with a plasma membrane, but because everything in the world of muscle physiology has its own special name, instead of the plasma membrane, you call the plasma membrane of a skeletal muscle cell the sarcolemma. But the sarcolemma is the same thing. It's a phospholipid bilayer surrounding the skeletal muscle cell. And then if you look inside the skeletal muscle cell, you see that it's made up of more cylindrical structures called myofibrils. And then if you follow the arrow to the right side of the screen, you can see the myofibrils in more detail. And if you remember from 201, skeletal muscle anatomy, when the early histologists took a micrograph of skeletal muscle, they saw these bands and discs and zones. So if you look at on the right side of the cartoon at the top, there's an I band and an A band. So if you look at the I band, you can see within the I band, which shows up as a lighter band in electron micrographs. Within the I band is a Z disc, so you see that sort of zigzaggy line called the Z disc. And then there's an A band, and within the A band you have the H zone, and in the middle of the H zone of the A band is the M line. And all these A bands and zones and lines and discs, they're just structures that you see on an electron micrograph, and they're made up of proteins. So if you remember from 201, the proteins in the sarcomeres, which is from Z disc to Z disc is one sarcomere. Those proteins, the major proteins, are actin and myosin and tubulin, and then there's one more that is the springy protein called titin. So let's see. So then in the cartoon, in the top cylinder, the light blue lines, that's actin, and then sort of the thicker orange lines overlapping the actin, that's the protein myosin. And then if you see that kind of light blue tubular structure on top of the second myofibril, just below the top one we were talking about, you can see a triad, a structure called a triad. And the triad consists of a T-tubule, and the T-tubule is just when the sarcolemma, or the plasma membrane of the muscle cell, dives down inside the cell itself. That's the T-tubule. And then on either side of the T-tubule are the terminal cisterns of the SR. The SR is the sarcoplasmic reticulum, and that's where calcium is stored. And then it says at the bottom right on the cartoon, that shows there's plenty of mitochondria in skeletal muscle cell, because you need to make ATP to contract the muscles. So at the bottom, I just reminded you that myofibers, that's a muscle cell, contain myofibrils. So in this cartoon, there's 1, 2, 3, 4, 5 myofibrils, and then the myofibrils are made up of myofilaments. That's actin and myosin myofilaments, and actin and myosin are the major proteins in the sarcomere, which is the contractile unit of the muscle cell, both for skeletal muscle and cardiac muscle. So this is a review of resting membrane potentials, RMPs, and APs, or action potentials. So if we look at the right side of the screen, we can see the phospholipid bilayer. and then there's some proteins inserted in the phospholipid bilayer. You have the cytoplasm in the lower part of the screen, and then the extracellular fluid in the upper part of the screen. And then you see all the ions that are in the extracellular fluid, ions that are in the cytoplasm or the intracellular fluid. So in the extracellular fluid, you can see there's an abundance of sodium ions. If you look in the intracellular fluid or cytoplasm, there's not many sodium ions. So there's more sodium on the outside than there is on the inside. If you look at potassium, that's K plus, of course. There's potassium both on the inside and the outside of the cell, but there's more potassium on the inside of the cell. There's also a chloride ion, that's Cl minus, and calcium, that's Ca plus 2. There's also on the inside of the cell, there's a big orange circle, well, kind of an oval with an A minus on it. That stands for big proteins inside the cell that contribute to the negative electrical charge inside the cell, but they can't leave the cell. So the proteins are too big to get through any of the channels in the plasma membrane. In this cartoon, it calls these channels in the plasma membrane potassium leak channels, but you know from 201 that there's not only potassium leak channels, there's sodium leak channels, and then there's the famous sodium-potassium ATPase, or pump. So at the cell's resting membrane potential, that's when the cell is not firing an action potential, but the cell is at rest. There's more sodium and calcium in the ECF and more potassium in the ICF. So remember, the sodium-potassium ATPase pumps 3 sodium outside the cell for every 2 potassium it pumps inside the cell. So there's more total positive ions in the ECF, and there's more total negative anions in the ICF. Calcium is also in the ICF, in the sarcoplasmic reticulum, but it doesn't contribute to the positive charge of the ICF because it's sequestered. Calcium is sequestered in the SR, or sarcoplasmic reticulum, so it doesn't contribute to the electrical charge of the cytoplasm or the ICF. And then this is just a action potential that you've seen from 201. If you look on the y-axis, it says membrane potential in millivolts. If you look on the x-axis, it's time in milliseconds. And then if you look at the resting membrane potential, it's where the line is on the left side of the cartoon, where it starts at minus 95 millivolts. That's the resting membrane potential of that cell for this action potential trace. Then it shows you that when the sodium channels open, sodium comes rushing into the cell, it makes the cell more positive. So you can see the electrical trace getting more positive. It's moving towards plus 30 millivolts. At plus 30 millivolts, the sodium channels will close and the potassium channels will open. And potassium will diffuse out of the cell to the extracellular fluid, and that's called repolarization. So basically, both sodium and potassium are going to move when channels are open, and these ions are also going to move because there's a concentration gradient. So sodium's going to move into the cell because there's a concentration for sodium. So there's a larger concentration of sodium on the outside than there is in the inside. And when sodium is moving into the cell via open sodium channels, that event is called depolarization. And then when potassium channels open and potassium moves out down its concentration gradient from high potassium in the inside of the cell to low potassium on the outside of the cell, that's called repolarization. And then when all the channels are back to the resting membrane potential, you go back to minus 95 millivolts. So this is just a review of excitation contraction coupling from the motor neuron to the muscle cell, and then the sliding filaments. So if you look at this cartoon, you can see this is a skeletal muscle, and you can see the myofibrils. It shows a sarcomere going from Z line to Z line. It shows the terminal cistern and the T-tubule, or the terminal cistern of the sarcoplasmic reticulum that holds calcium, shows the T-tubule. Remember, the T-tubule is where the sarcolemma dives down into the inside of the muscle cell. And also remember, when you have the T-tubule and the terminal cistern together, or the terminal cistern of the SR together, that's called a triad. And then you see the yellow structure. That is a neuron, of course, and it shows that the bulbous part of the neuron is called the axon terminal, and that's where when you want to contract your biceps muscle, say, you fire an action potential from a motor neuron in your spinal cord, travels to your biceps muscle, and the electrical signal at the axon terminal will cause the release of acetylcholine into that synaptic cleft, so that cleft between the axon terminal of the motor neuron and the sarcolemma of the skeletal muscle. Acetylcholine is released, binds to acetylcholine receptors in the sarcolemma of the muscle cell, and an action potential then is generated. And then the action potential can travel along the sarcolemma. And you can see that pink line is the action potential. The action potential dives down into the T-tubule, and then that action potential, remember, it's an electrical signal, so it can open voltage-gated channels in the sarcoplasmic reticulum, causing release of calcium, and then we'll talk about what calcium does to issue a contraction of the sarcomere. So it says on the left, I've got some notes. It says the heart muscle does not need a neuron to contract, but it is innervated by the sympathetic nervous system, or the SNS, and the parasympathetic nervous system, or the PNS. So heart muscle can contract on its own, as opposed to skeletal muscle that cannot contract on its own. A skeletal muscle needs a neuron to tell it to contract. The heart muscle does not, but the heart muscle contractile strength can be modulated by the sympathetic and parasympathetic nervous systems. So this is just a cartoon. Again, this is a review from 201. If we start at number 1 at the top of the screen, we can see that the orange structure with the little golf club type thing on it is myosin. So that's a myosin cross bridge. That's a myosin head binding to actin. So the myosin head is binding to myosin binding sites on the actin filament, which are those little blue dots with the little black space in there. At that point, calcium is present because, remember, an action potential was fired. The SR released its calcium. Calcium then bound to those little yellow structures, the troponin-tropomyosin complex. And when calcium is present, then troponin-tropomyosin complex, that yellow thing with the sort of orange rope on it, can move out of the way of the myosin binding sites on actin, and myosin combined. So you can see on the myosin head, they're calling it the high energy configuration because it's bound, the myosin head is bound to actin, and ADP and inorganic phosphate are still attached. So just previous to that event, ATP was hydrolyzed. So if we look at step 2, they call it the working stroke. So it's working because the myosin head is pivoting, and this is going to cause the sarcomere to shorten, and with all millions of sarcomeres shortening, then your biceps muscle will shorten as well. So in the working stroke, ADP and inorganic phosphate are released, the myosin head pivots, then from step 2 to step 3, ATP comes in and binds to the myosin head. Again, when ATP can bind to the myosin head, then that allows the myosin head to release from binding to actin. So you need ATP present so that the myosin head can release from actin. And then from step 3 to step 4, ATP then is hydrolyzed. So that means the inorganic phosphate is split. A single inorganic phosphate is split off ATP. So then you have the inorganic phosphate and ADP left. And at that point, the myosin head cocks back into position so that you can you can cycle through another event of myosin head binding and releasing.